Thank you very much. Good afternoon. Assalamualaikum warahmatullahi wabarakatuh. Professor Badrul Hegar as the Director of IMERI, Dr. Diki Tahapari as the Chief of the Metabolic, Cardiovascular, and Aging Cluster, IMERI, and also the Dr. Ippei Shimizu from Jutendo University, Tokyo, Japan, and also Mrs. Rafika from the Chemical Medicine, Faculty of Medicine, University of Indonesia. So today we would like to perform the initial innovation in aging science, or the IAS, in the webinar series 1. So in this topic will be the anti-aging that eliminates the senescent cell. So hopefully this IAS webinar series will be performed every 2 months, and we will also perform another webinar series maybe in the April, and also maybe in the June also. Okay. Before we start the discussion here, so it's very pleased that we have Professor Badrul Hegar, specialist and consultant, as the director of the IMERI, and we would like to give some to ask Professor Badrul Hegar for giving some opening statement. So please, Professor Badrul Hegar, the time is yours.
Okay, thank you very much, Dr. Agi. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, all. Of course, the Honorable Dr. Ippei Shimizu, Ph.D, FESC, FAHA from Jutendo University, Tokyo, Japan, and also Rafika Indah Paramita, Master of Pharmacy, Apotheker. She is a scientist and researcher from Bioinformatic Core Facility IMERI FKUI, and also Dr. Agian, Jeffilano, Barinda, Ph.D, as the moderator, is also from Metabolic Disorder, Cardiovascular, and Aging Cluster IMERI FKUI. Once again, I would like to appreciate for MFA cluster for the conducting such interesting event this afternoon. Yeah. I think this kind of event is, of course, very important for IMERI FKUI, especially for our scientists and researchers, to provide their inspiration in developing research and innovations. If we look at the topic is really very interesting and challenging issue. Yeah, anti-aging vaccine that eliminates senescent cells. As we know that senescent cells refer to those that hope stop dividing but do not die. They damage nearby healthy cells by releasing chemicals that cause inflammations. Currently, I saw in the literature, the Japanese researcher has developed a vaccine to remove senescent cells that accumulate with age and damage nearby cells, causing aging-related disease. Right now, also, many of the existing drugs to remove senescent cells are used as anti-cancer agent and may cause negative side effects. So we hope that the side effect from the vaccine that will be developed were fewer, while its efficacy lasts longer as well, of course. We also expect that the vaccine will be applied to the treatment of aging-related disease, such as arterial stiffening or diabetes or other else. And I hope at this scientific event this afternoon, we will have the knowledge regarding the vaccine work and also the experimenting with senescent cells as well. I hope this topic and discussion will become an idea for the scientists and also the researchers to create a better quality of healthcare and also quality of life through innovation in medical research to the society as well. I would like to thank very much to our guest expert, Dr. Ippei Shimizu, of course, not only for this afternoon, but also for the collaboration that has been established, especially for our scientists and researchers from FA Cluster IMERI FKUI. Okay, please join this scientific meeting. I hope we get a lot of knowledge from this event. Thank you very much.
Okay, thank you very much, Professor Badrul Hegar, Ph.D. Specialist and Consultant, for your wonderful opening statement. Okay, and then we can move on to the next session. So the next session. Okay, the next speaker will be the Mrs. Rafika Indah Paramita, Magister Pharmacy, Apotheker. So as the brief introduction, she was graduated in 2014 for the Bachelor of Pharmacy at Erlangga University, and then continued to study the Master of Pharmacy at University of Indonesia in 2017. And currently she is the lecturer in the chemical medicine in the Faculty of Medicine, University of Indonesia, and also the researcher in the Bioinformatics core facility. She is the expertise in the epitope vaccine. So therefore, today we are very pleased to invite her to give the lecture with title The Reverse Vaccinology in Peptide-Based Vaccine Design. So please, Mrs. Rafika, the time is yours.
Yeah, thank you very much, Dr. Jeffy. Good afternoon, everyone. Thank you for having me today. Before I start, I want to greet Prof. Hagar as our director of IMERI, and also Ippei Shimizu Sensei, and also Dr. Diki. Thank you. And also good afternoon for all of the participants. Thank you for joining our event today. Okay. Yeah. Today I would like to present about a reverse vaccinology for peptide-based vaccine design. So it's more from a bioinformatics point of view. So maybe I'm not really related with the topic. So maybe it's more general for peptide-based vaccine design. Okay, here are my outlines. I will little bit talk about human immune system, and also the vaccine science using vaccinology, and the example of the peptide-based vaccine design. Okay, human immune system consists of the innate immune system and also adaptive immune system. The adaptive immune system response is antigen-specific immune response is more complex. So I'm not really, I will talk about innate immunity, but only on adaptive immunity, and especially in T cells response. The T cells and B cells are the major cellular components of the adaptive immune system. And we can also split the kind of adaptive immunity as a humoral and also as a cell-mediated immunity. Immunological memory is developed through memory B cells and also memory T cells, resulting in enhanced response to subsequent encounters with the same antigens. For the first one is humoral immunity. The humoral immunity is mediated by B cells, and the cells-mediated immunity mediated by T cells. They work closely to combat infection in antigen-specific. In this picture, I have example of the humoral immunity. So B cells will produce antibodies to eliminate extracellular mechanism to prevent the spread of infection. So the antibody will recognize the antigen just like lock and key concept. So the antibody will have the lock, and the surface on the antigen on the surface of bacterial cell or on a virus and a surface will as the key. So it will match the key lock and key concept in here. And from the cell-mediated immunity, the MHC or major histocompatibility complex protein play a vital role. T cells identify the foreign antigens through their T cell receptors, which interacts with MHC peptide complex. So the peptide will bind first to MHC, and after that will bind to TCRs or T cell receptors. For the MHC class 1, the antigen will process the pathway. It includes the first step is the proteasome, and here we'll chop the protein into shorter peptides, and then the translocation of the peptide into ER by protein. And then after going to ER, the peptide will bind to specific MHC. If in here you have MHC class 1, the specific peptide is 8 to 11 amino acid peptide. So for MHC class 1, it will bind to shorter peptides, only 8 to 11, to binding group of MHC molecules. And after that, they can move to cell surface and will recognized by CD8 plus T cells. How about the MHC class 2? For the MHC class 2, they will bind to endogenous cellular protein or peptide that typically longer than the in MHC class 1. So in MHC class 2, they can form like 8 to 30 amino acid in length. And after they bind to MHC class 2 protein, they will recognize by CD4 plus T cell protein. The peptides that are presented by MHC and also recognized by T cell are termed with T cell epitopes. So in simple terms, T cell epitope is like a peptide antigen that can bind to MHC or major histocompatibility complex, and also can bind to T cell receptors. So you can say like just you can say that was T cell epitopes. So if the peptides antigen only can bind to MHC, it can say as T cell epitopes. So it's only MHC binders, something like that. The T cell epitopes are important targets of adaptive immune response and rational vaccine design. This is a simple picture of the epitopes. So we have antigen in here, and then we'll chop into short peptides. The peptide will bind to MHC, and if in the cell surface, if they can recognize by T cell receptor, they can become an epitope. Talk about immune response system. Of course, we will talk about MHC or HLA in human. So it's same but a different term. In human, we call it by HLA, leukocyte, and so it's a human MHC. It's located on chromosome 6 on the short arm. And these HLA genes are most diverse number of 2021. HLA have more than 32,000 allele. So you can check to this website, and they I'm not if I'm right until December 2021. This HLA regulate immune responsiveness and also play a vital role in regulation of cell-mediated immune response. Okay. Vaccine research nowadays there a lot of advanced in biomedical technology, and also the bioinformatics tools that can let the generation of large amount of data and information useful for vaccine research. If we talk about research, maybe in a conventional way, the first step is always about the experimental or wet lab research. But because of we have already had a lot of data, a lot of database, we can use the previous data to design first. So it's like a prediction or in silico first. Yeah, we can predict we can design first the vaccine, predict how the immunogenicity, how the coverage, something like that, before we start on experimentally wet lab. So because of we could predict, of course, we will need the computational approach, and this computational approach include the collection of data, manage the data, and also mining the data. Because if we all know that genomic data is like that kind of huge. Yeah, we have like big data on the genome data. So we really need computational approach because we can't handle it manually. So we need the help of the computational approach. So because of that, because we reverse the experimental way, we first collect the data first, and then we do a prediction first. After that, after we get the rational design, we can move to the experimental research. So because we reverse the methodology of the design vaccine, we call it reverse vaccinology. There's like vice versa from the conventional vaccine development. Maybe the first way on conventional, we like trial and error in wet lab. We use microorganism. We sequence first. We get the antigen, and then we clone the genes, and we try to develop the vaccine. But because of right now we have like a sequencing machine. We have a sequencer. We can know about the sequence of the antigen, and we can know the sequence of the pathogen. Yeah, it's make us easier to know which one of the peptide or the pathogen that have immunogenicity that fit most. So the step for reverse vaccinology, the initial experiment, we of course we need the sequence data. We can find this data in a lot of databases such as NCBI or EBI or BDBJ. Yes, such a lot of database that we can use to generate to become our data set. And then next to computational steps, we collect the data, and then we did some prediction of vaccine candidates using a lot of computational tools. And right now, there are a lot of computational tools that we can use. Maybe in the next session I will explain more. And after we get the vaccine candidates, we can move to experimental steps. Yeah, one of the difficulties of design vaccine is due to diversity. What I mentioned before, HLA genes is one of the most diverse gene in human. So because of the diversity, it's a little bit difficult to make a vaccine that fits to a lot of population. But the advantage of the diversity, no single pathogen can wipe out the population, because this is like example, me and Dr. Jeffy, for example, have a different HLA allele, for example, and because of we have a different allele on HLA, we have like different immune response, something like that. So although it makes difficulty to design vaccine, the diversity will provide protection and position to development for vaccine effective at the population level. When talk about HLA, actually HLA allele can grouping to some supertype. We term it with supertypes because a lot of allele like really different each one to another. So allele can have like a similar or overlapping peptide binding specificities that make they can group into one supertypes. And these supertypes are useful for defining promiscuous peptide that are able to bind to numerous allele. So from the previous research, if the epitope can bind to more HLA supertypes, the coverage of the supertypes will get increased. So if here, you can see here, if the epitope can bind to HLA supertypes like A2, A3, and B7, the average of the coverage is about around 86.2. But more epitopes can bind to more HLA supertypes, the average of coverage population will increase around 99.8. So it's important for the researcher that have design vaccine research to make vaccine design that especially for peptide-based vaccine to design the promiscuous peptides. This is the HLA supertypes groups. So in here we have HLA-A, A1, A1A3, A24, A2, A3, and A24. And this is for HLA-B supertypes. There are B7, B8, B27, B44, B38, and B62. And in each HLA supertypes, they have a lot of HLA allele. So yeah, you can see here that maybe this one and this one have like a similar binding sites, binding motifs, something like that. Yeah, this is like I previously mentioned about promiscuous peptides. So if we design the peptide-based vaccine design, we need to design the promiscuous peptide. What is the promiscuous peptide? So promiscuous peptide is a peptide that can bind more than one HLA variant. So the peptide can bind to more than one supertypes, it will increase the population that I mentioned before. So it's like different with the precision medicine. So if precision medicine, we take a look to like one population into different drug for different populations, something like that. But for vaccine, we try to make like one vaccine that fits for all. So it's important to design the peptides that can disperse. So why we need the computer models? So only 1% to 5% of peptides from a protein can bind to HLA molecule. So like I previously mentioned, that only like 8 to 11 peptides can bind to HLA or MHC molecule. So it can be random way. So if we use the computer model, we can predict which peptides that can bind specifically to HLA. So if we use the traditional ways, kind of slow and also take a lot of time, take a lot of cost. So with the computer modeling, it enables systematic screening for HLA binders, minimize the number of experiment, and also diminish discovery cost by time. So this is like example, if we did the experimental way to have a T-cell epitope. So this is for example, if I have a peptide that have 495 amino acid long, it will chop into 8 to 11, 8 to 11 amino acid, or we just about get example on 9 MERS in here. So we chop into 9 amino acid. So we'll get 487 IMERs. And if we try to assay the peptide to HLA molecules, for example, there's 12 HLA molecules, so it will get 6,000 binding assays. So it really a great big number for assay. So for to help the researcher to simplify the experimental way, we can predict use the computational approach, and we just take a look which peptide that have good like interaction with HLA molecules. So we don't have to say a lot all of these peptides. Yeah, this is the example that I will show in cancer vaccine. Yeah, cancer vaccine actually have the same principle with another vaccine or example from the virus or bacterial vaccine. So the cancer antigen will recognize as the foreign or as the pathogens. One of the hallmarks of cancer is its ability to avoid immune system. And the cancer vaccine requires the strong activation of the T cell adaptive immune response to elicit their anti-tumor potential. And the aim of cancer vaccine is strongly activate the CD8 plus T cell and mediated by CD4 T cell. And can overcome self-tolerance and immune suppression, leading to elimination of cancer cells. In cancer vaccine, we design the TAs or tumor antigens. So tumor antigens is tumor-derived molecules that interact with cells and products of the immune system. And the tumor antigens can be subdivided into two main groups. The first one is tumor-specific antigens. It's like exclusively found in tumors, and we can find in a healthy human. So we can say that's neoantigen or cancer test antigens or uncovered antigens. And also we have tumor-associated antigens that often expressed in tumor cells relative to their healthy counterparts. For example, in breast cancer, we in TAs like the offer express of the HER2 protein, for example. And the new antigens are newly formed protein antigens, either from somatic mutation or the viral genes that get incorporated in infected cell genome. And in recent years, the therapeutic focus has shifted away from the TAA to the use of TSA. So especially we try to design based on the T-cell neoepitope. Yeah, this is like one of the workflows. So it's not only a way to do the peptide-based vaccine design. A lot of workflows that we can use, but that's we can use more or another software to use for the design. And for this design, I collect the sequence from the TP53 protein that appear in breast carcinoma tissue. Why I did this? Because in TP53, there are a lot of mutation that, that appear in breast cancer, breast carcinoma. So I'm not designed based on the wild type sequence, but I tried to find the epitope in the conserved sequence. So I don't want the diverse sequence that maybe come as the potential epitope, but I focus only on the conserved sequence. So I collect the mutant protein. I collect from Cosmic, and I only look on the missense mutation only, and it generate around 2,000 sequence. And after that, I will remove the duplicate or identical sequence. So I only got the 175 sequence as a unique sequence. So I will proceed only the unique sequence. For the duplicate or identical sequence, I will not proceed further because yeah, they have same sequence. So it will, if we also include that on the analysis, it will will have the process of analysis. So for after that, I analyze the diversity of the mutation using AVANA or antigenic variability analyzer tool. So in with this software, it can chop the protein into 9 mers or 9 amino acid peptide, and which peptides that conserve, which peptides that diverse. So from this analysis, I got around 157 conserved sequence peptides. So it means conserved, there's no mutation that found in these peptides. And after that, I check the interaction between the peptides with the HLA supertypes. I use in here that I use that CTL that you can find in this website. And from this analysis, I only got 8 putative promiscuous HLA. So from the 157, only 8 peptide that found promiscuous or can bind to a lot of HLA subtypes. And after that, I can also predict the immunogenicity, use immunogenicity parameter in IEBB. And also I can check the if there are any previous research that already say the MHC and ligand in immune epitope database also or IEBB. And we can also check the toxicity and also the allergenicity. For toxicity, we can use toxin PrEP, and the allergenicity we can use AllerTop version 2.0. Yeah, this is the result for entropy analysis. In here, we can have this plot from the AVANA software. We can see in here just there is some position in the TP53 protein. If there's no peak in here, so it means the sequence are conserved. But if we got a high peak in here, so it means the peptide around this is very diverse. So I will remove the peptide that show high diversity. I only take a look to peptide that conserve, so no peak from the AVANA result. Yeah, I did the HLA supertypes prediction through NetCTL. And this is the website, and there's a lot of class 1 supertypes in here. And this is like the results. So if the peptide is found have interaction, they will give the -E symbols on here. And this is the IEDB website. IEDB have a database and also the resource for analysis, as I mentioned before. And from this research, I get 8 putative peptide epitopes that combine to HLA supertypes, and this is the immunogenicity prediction. And we can also check the previous research that maybe the peptides can bind to MHC ligand assay. So this is already done before in previous study. But like, yeah, not close, not exact epitope they use, but yeah, some part of them already say through against the HLA. From here, we can see that only 4 epitope, only 4 peptide that they already test before, that exactly same with that I predict with the workflow. And we can also check the allergenicity and toxicity. And also there's a software that called Tantigen 2.0 that can comprehensively predict the epitope. So we just put the sequence and they will do the rest. So we don't have to did one by one, just like I did before. So they will provide a lot of T-cell epitope and lines in here. G-binders that I use the term before. Okay, the last summary, the T-cell epitopes are important targets of adaptive immune response and national vaccine design, and computational tools and database play a role in support of developing vaccine design, especially in vivo vaccinology. So next generation sequencing that provide the sequence DNA, HLA allele diversity, utilization of T-cell epitope prediction, and analysis of HLA peptide binding algorithms will help the researcher to design rational peptide-based vaccine. So this is the acknowledgement from my professors, Prof. Muhammad Asifan and Prof. Guang. So yeah, that's all from my presentation. Thank you very much for your attention. Good afternoon. I give back to Dr. Jeffrey.
Yeah. Thank you very much, Mrs. Vika, for your wonderful and very advanced lectures. Yeah, I really apologize that I forgot to take a picture, to take a photo for this webinar. So maybe before we continue our session, so please kindly to turn on your video and then we can take the photo together. Okay. Yeah. Yeah. Okay. Can maybe the Miss Tika or Mr. Budi to give to take a photo.
Okay. Baik dok, saya izin untuk screenshot ya. Baik, untuk halaman pertama, 1, 2. Untuk halaman selanjutnya, 1, 2. Sudah dok.
Okay, thank you very much, Ms. Tika. And also we will go into the next discussions after hearing the basic or the overview of the epitope vaccine. Now we will hear the lecture from the Dr. Ippei Shimizu. So as for his background, he was graduated as the medical doctor in 2002 in Chiba University, and also continue to study in doctoral course in also Chiba University, and also the postdoc in the Boston University. And currently, he is the associate professor in the Department of the Cardiovascular Biology and Medicine in Jutendo University Graduate School of Medicine, Tokyo, and also the member of EMET for project for elucidating and controlling mechanism of aging and longevity from Japan Agency, and also the leader of the U45 International Society of Heart Research Japanese sections. So he was published several interesting articles, particularly the senolytic vaccine articles. So today, we will hear about the senolytic vaccine. Please, Dr. Shimizu, the time is yours.
Hi, Jehi. Thank you very much for your kind introduction. And I would like to thank Jeffy and the organizer for giving me these great opportunities. So you see what I see now?
Yes, please.
Okay, great. Okay, so we are I'm working in the Department of Cardiology, but our research topic is aging. So with aging, there are several machineries that are known to associate with biological process. So for example, mitochondrial dysfunction, stem cell exhaustion. So we extensively studied the role of cellular senescence in aging and age-related disorders like heart failure, obesity, diabetes, and atherosclerotic disease. Okay, so this is the summary of my the publications. So the keyword would be adipose aging, for example. This promotes pathologies in heart failure by introducing like hyperinsulinemia, the systemic insulin resistance. Also, this holds true also in the obesity and diabetes. Also, we are interested in the role of brown adipose tissue in heart failure and obesity and diabetes. Also, the bone marrow aging and cell aging has a role for heart failure and obesity and diabetes. So it sounds that the aging has a kind of may break as kind of a breakthrough to comprehensively understand these age-related disorders. Okay, so this is the review article for Dr. Skonye, published in 2015. So with aging, the senescent cells make a kind of accumulation, and then the senescence associated secretive phenotype, like a chronic steroid inflammation, contributes for the tissue remodeling. And for example, like insulin resistance or like a chronic inflammation promotes pathologies in obesity, diabetes, heart failure, and atherosclerotic disease. So there are two types of cell senescence. So one is described as replicative senescence. So every time when the eukaryotic cells make a proliferation, the telomere make a kind of alteration, so they become short. Then after reaching the critical threshold, this is recognized as a DNA damage response, and then P53, P21, or P16 pathway promotes cell to senesce. Also, other cell senescence is termed stress-induced premature senescence, when the cells are exposed to oncogenes like excessive reactive oxygen species, cytokines, genotoxic agents, again P53, P21, or P16 pathway promotes cells to senesce. So the characterization of cell senescence is the termination of proliferation, and also the genetic profile changes dramatically. So they become like a pro-inflammatory phenotype. So this is an interesting paper showing the bystander effect. Okay, so these cells are young cells. So this cell is an age cell. So when the age cell make a deep dive into the cell, a young cell population, even these young cells make a cell senescence. So this arrow indicates the DNA damage markers, the gamma HCAX. So when they make a kind of a direct contact, these cells become age have a kind of a senescent phenotype. So this is described as bystander effect. Going back to 2009, with Professor Minamino Toru, we show that the obese metabolic stress, so oxidative stress, DNA damage, telomere dysfunction develops in the visceral adipose tissue that promotes the increase in P53. And then P53, the excessive signal pathway introduce pro-inflammatory cytokine production, systemic insulin resistance, and diabetes. This was highlighted as adipose tissue aging. This is a study from humans, like a non-diabetic patients. The type 2 diabetic patients in the adipose tissue show an increase in P53, and also the senescence associated beta-gal positive cells increase in the adipose tissue, and also markers for cell senescence like TNF, CCL2, and P21 increase in diabetic patients. So we also analyze heart failure mood models, so mice models, and we found that like similar with the obese model, we found that the left ventricular pressure overall introduced chronic steroid inflammation in the visceral adipose tissue, and also increase in P53 in the adipose tissue. And then this led to the chronic steroid inflammation in the visceral fat, and then there's the production of pro-inflammatory cytokines, including TNF, and then insulin resistance and hyperinsulinemia. Insulinemia develop under this condition. I will move. All right, I will go back to our. Also in the aorta, so this is a dietary obese model, and then in the end cells of the aorta, it's a little bit difficult to see the signal, but this brown signal indicates P53, and also from the MAC study, like CD45 negative, CD31 positive cells, like mostly considered to be like endothelial cells in the lung and muscle, they become high positive for P53, as you can see here. And we found that the metabolic stress increased P53 in the endothelial cells. This is a reduction in the phosphoinos, also GLUT1 expression in the skeletal muscle, and then these leads to the reduction in mitochondrial biogenesis and glucose uptake, leading to systemic insulin resistance. Also, when we make a deep dive into the P53 expressing cells in the failing heart, we found that during the left ventricular pressure overall model, the endothelial cell P53 became high, as you can see here, within the staining and also the FAHA study. So like heart failure and obesity, they're like a common pathogenic pathway. The endothelial cell P53 expression increase, leading to cardiac inflammation, promoting pathogenesis in heart failure. And at the meantime, in the obesity, endothelial cell P53 promoted systemic insulin resistance. So the question would be whether the suppression of P53 becomes a therapy itself. But as you know, the P53 is a kind of a good guy in the field of cancer. It's described as a guardian of genome. If the patients have a mutation in P53, then of course the issue would be like it would increase the risk of tumorigenesis. So as many of the attendees audience knows, so I would like to share what's new in aging research. Okay, so this is a review article from Nature published in 2019. So, all right, so we are here, like the term senolytics. As you know, this means a specific depletion of senescent cells. So this is even tested in humans by Cochran in Mayo Clinic. Okay, so I brought this figure from the recently published review article. So this is a characterization of senescent cells. Compared with a young cell, they become enlarged and they become flat. Also, they show secretory phenotype like interleukin 6, MCP1 is produced or described as senescence associated secretory phenotype SAS. Also, they show a high expression profile for P53, P21, P16, and also they have a high profile for DNA damage response like gamma HAX, P38 MAPK. Also, they show an increase in the SA beta gamma signal, and also they have a telomere attrition, as you can see here. Also, some people mentioned that the TAF, telomere associated foci, defined as telomere signal together with an increase in the DNA damage response. So, for example, in the cardiac tissue, TAF is recognized as the marker for cell senescence, the presence of cell senescence in cardiac tissue. Okay, so the recent study have been like a kickoff from Dr. Karkman's lab. The point is that nowadays it seems technically possible. We're still working on it also to deplete the target these senescent cells. Okay, so this is a genetic model of P16 depletion model from Van Duersen's lab. Okay, so this is a progeria mice. So in the control mice, so this is a progeria, and then this is a P16 depleted progeria mice. So morphologically, you can see they show all kind of a healthy morphology. It's almost like unbelievable to believe that these mice are in the same age. So they show a reduction in the scoliosis, and also the level of the incidence of a cataract will be diminished, as you can see here. And also, this is this paper analyzed a different P16 depletion model and tested the phenotype in atherosclerosis. Okay, so after the administration of a drug, then the P16 positive cells may be depleted, and then in the genetic model of the atherosclerosis, the level of atherosclerotic plaque will diminish, as you can see here. Okay, so those two papers tested the transgenic mice system that enables the researchers to specifically deplete P16 positive cells. And this interesting paper showed that the ABT263 it's kind of an anti-cancer drug, so we never feel free to take it. So, but this also has a kind of we characterize as a senolytic agents. So senolysis means the specific depletion of senescent cells. A senolytic indicates the agents equipped with the capacity to deplete, the mediate senolysis. So ABT263, after the administration in the young cells, in the high concentration, the cells continues to be viable, but in contrast, in the aged fibroblasts, the viable cells are started to reduce after the administration of high concentration of ABT. So through this study, ABT263 is recognized to mediate the senolysis. Also, they tested the muscle stem cells after the administration of ABT, the marker for cell senescence, and also DNA damage response are diminished. And also, these stem cells are started to reproliferate, enhance the proliferation. Another study tested ABT263 in the myocardial infarction model. So after the myocardial infarction model, some group were introduced with nabitocrox, the different name of ABT263. And as you can see here, after the administration of ABT263, even it's improves the survival rate after myocardial infarction. And also the ejection fraction seems to be improved by the nabitocrox administration. Okay, so this is a summary of the figure we made in 2018. At that moment, there are 14 senolytics. Dasatinib and quercetin and nabitoclax are extensively studied. And also, even in the ongoing clinical trials were performed with dasatinib and quercetin administration. But as you know, okay, so the D and Q are tested in humans in the chronic kidney disease, idiopathic pulmonary fibrosis. But as you know, the dasatinib is a tyrosine kinase inhibitor, quercetin a flavonoid group of polyphenol. ABT263 is a BCA2 family inhibitor, so it's kind of anti-cancer therapy. So we may feel free to take quercetin because it's kind of polyphenol. So we feel free to take it. But the point is that whether there will be less toxic reagents, senolytics. I mean, we can feel free to take in our daily life. Okay, so we are exploring new senolytic agents. So one is one of the drug that we use in the real world, and another will be a herb that we also feel free to take in our real world. I mean, in the daily life. Okay, so the high-fat diet will increase SA beta cells in the fecal adipose tissue. Administration of Steno 7284, the signal will diminish. Also, DHT signal, this is a reactive oxygen species, will increase in the fecal adipose tissue after dietary obesity, and also this drug leads to the reduction in the reactive oxygen species in fecal adipose tissue. Also, P53 signal will enhance with a high-fat diet, and then reduce with the Steno 7284 administration. There's a high-fat diet leads to an increase in the chromosomal structure. So this is the hallmarks of on the mononuclear cells, most like macrophage. Also, co-administration of Steno 7284 leads to a reduction in the infiltration of the mononuclear cells. Also, on the system glucose intolerance will improve with Steno administration. Also, in the up, we knock out mice subjected to Western diet. So you can see that SA beta-galacina will enhance like this. Steno administration leads to a reduction in the signal. And also, the atherosclerotic plaque looks reduced with the Steno administration. So this is Goro Katsumi, who is extensively studying this research project. Also, we are interested in the herb because it's a kind of food material, so it's less toxic. So ABT263 as a positive control, administration of the ABT as previously reported reduce the viable cells in the senescent cells. Okay, so these are Hubeck, the M cell cells. Also in the Seno-H, Seno-Herb, so we cannot disclose at this moment. So it's a high concentration. It sounds no effect for the viability in the young cells, but in the senescent cells with a high concentration, they become less viable. And also compared with wild-type mice under dietary obesity, the administration of Seno-H leads to a reduction in SA beta signals you can see here. Also, we are testing the activity of the mice. Administration of Seno-H enhance the activity analyzed under the open field test. Also, the rerunning test show the enhancement in the activity of the mice. And also, it doesn't reach statistical significance at this moment because of the rather limited number of mice analyzed. By some, they show kind of a trend to increase the running distance, as you can see here in the treadmill examination test. So this is Ryo Furuichi, one of our lab members in Jutendo. Okay, going back to the Kirkland's human studies. So this is the report 2019, like two or 3 years ago. So they administered like D and Q, dasatinib or quercetin in humans, and found that P16 level was reduced, and P21 cells also reduced in the subcutaneous fat, and also the SA beta gal cells are reduced in the subcutaneous fat. So this indicates even in humans, the DNQ treatment mediates like a senolytic effect. And also going back to the target, so we recently identified a GPNMB as a cell surface marker for senescent cells. Analyzing two subsets of transcriptome studies, we got interested in the transmembrane and GPNMB glycoprotein non-metastatic melanoma protein B having a transmembrane domain, showing an increase in the transcript level with cell senescence. In the HUVEC, we found that senescent cells exhibit high expression profile for GPNMB in protein, as you can see here. Also, transcript GPNMB show an increase, as you can see here. Also, we analyzed aorta from the aged mice and found that GPNMB expression together with other senescence markers showing an increase in the aorta. And also, the transcript GPNMB show an increase in the aorta, as you can see here. We also analyzed the ApoE knockout model and found that the protein GPNMB together with other senescent markers increased in the aorta. And also in the transcript study showed an increase in GPNMB, as you can see here. We also tested the level of GPNMB in human aorta in atherosclerotic disease against non-atherosclerotic disease patients, and found that the AD patients has a high expression profile for GPNMB in the endothelial cells in aorta. Also, we did some FAHA study in the obese mice on the high-fat diet, and we found out that when they so this spiral beta gal stain is a commercially available kit, and this become positive like a SA beta gal a kit that can be used in the FAHs. So it's very convenient. So the senescent cells become positive for spiral beta gal signal, and these senescent cells, like 90% of them become positive for GPNMB. So this indicates that the senescent cells have a high expression profile for GPNMB, and we tested under the obese condition. So we generate the transgenic mice that enables to show so this is a GPNMB positive cell elimination mice. So the high-fat diet mice, they become high expression, they show a high expression profile for GPNMB together with luciferase. So these are GPNMB high cells. And also together with diphtheria toxin, we can deplete GPNMB high cells, as you can see here. So after diphtheria toxin administration, the SA beta gal signal will reduce, as you can see here. And also, of course, the GPNMB level will reduce with a DTR administration, together with a reduction in the senescent markers like P16 or P21, P16. And importantly, like a systemic glucose intolerance, as well as insulin tolerance tests, systemic insulin tolerance were ameliorated with the depletion of the GPNMB positive cells. And also on the diphtheria administration, a later reduction on atherosclerotic plaque, as you can see here in the ApoE knockout mice. Okay, so through the client collaboration with Dr. Hironori Nakagami in Osaka University, he was kind enough to give us some design the vaccine, the peptide vaccine for GPNMB. So this is a GPNMB conjugated with KLH, and also together with administration of the adjuvant like Titamax Gold. So we can the endogenous immune system will target the GPNMB start to produce, the B cell start to produce the antibody against GPNMB. So I'm not sure how he designed this, but like a previous presentation from Dr. Fika showed, he might have used some of those internet sites available, but I don't know. He may feel free or he may not feel free to share it with us. But anyway, he was kind enough to design us the GPNMB peptide function. Okay, so we use P19 of TTR mice. So P19 is also another marker for cell senescence. So these mice, when the P19 is positive cells are accumulated, they become positive for loose fetus. And then the GPNMB action reduces signal. And also the SA beta gas signal will reduce, as you can see here. And also the atherosclerotic plaque will reduce. And also the loose fetus activity will reduce, as you can see here. And also the glucose tolerance and insulin tolerance tests are ameliorated with GPNMB vaccine therapy. And then we administered these to the progeria mice, and we found that the lifespan does not change, but the median lifespan seems to increase in the somehow in the male mice, but not in the female mice. So there is a gender difference. So we're still working on it, but it's kind of a very interesting finding. Okay, so maybe a little bit time is up, so I need to make a rush. So we analyze the obese visceral fat and the GPNMB high cells. The CD31 positive GPNMB high cells were depleted with GPNMB vaccine, and CD31 negative, CD45 negative GPNMB high cells were depleted with GPNMB vaccine. These cells seem to be mostly fibroblasts. Also, we analyzed the aorta of the vaccine group and found that there is a little bit difference between the obese mice. Okay, so CD31 positive cells showing reduction in the GPNMB high cell, CD45 positive cells showing reduction in GPNMB high, and in the case of aorta, it seems that the GPNMB high macrophage seems to be reduced mostly by the GPNMB vaccine therapy. Okay, so we also analyzed the GPNMB knockout mice and found no change in the vaccine therapy. So if they don't have a expressed GPNMB, they don't mediate any biological effect, as you can see here. Also, we did some neutralizing antibody study, and in the control group, the beta gas signal was reduced with GPNMB vaccine. But if he introduce a NAB against natural killer cells or CD8 cells, this reduction seems to be the gain signal, as you can see here, and also the quantified data here. So this GPNMB vaccine, they have an ADCC assay, ADCC activity. So administration of this vaccine would promote ADCC activity and then kills the GPNMB high cells. So that's the system working in our model. Also, we compare with other senescent senolytics like DNQ or ABT. So there is no change. DNQ, nabitoclax, and GPNMB vaccine, no change in body weight, food intake, energy expenditure, no change. Interestingly, the DNQ, also nabitoclax and GPNMB, also this is the P19 loose fetus mice that show the reduction in the early phase after the administration. Somehow the signal gain, as you can see here, and after in the chronic phase, still the GPNMB vaccine group it sounds that the senescent cells continuously suppress compared with DNQ and the nabitoclax administration. And also the insulin tolerance tests were ameliorated with continuously ameliorated with the GPNMB vaccine group in the chronic phase. So we also tested the level of white blood cells. They show the reduction. So this is a potential side effect of the nabitoclax. But this doesn't reduce with the GPNMB vaccine therapy. So it sounds that they're more safe. The safety issue seems to be more reduced compared with the nabitoclax administration in the case of GPNF vaccine therapy. Okay, so this study was done by Masayoshi now in Mayo Clinic, working with Kirkland's lab. Okay, so I didn't have any time to talk about xeno-Qs suppression. We are also interested in xenometabolite or xenoprotein that increase with aging. Also, a suppression of these will be one approach. Also, the specific depletion of the age cells like xenolysis. This is another approach. And then this is a rejuvenation. Okay, so I think there are several other senolytics everywhere in the world. And it continues to be like an interesting research topic to explore, like penicillin found by Alexander Fleming. Okay, so cellular senescence promotes pathology in the age-related disorders, and senolysis reverse aging phenotype. And senolysis is a promising approach to combat cardiovascular and metabolic disorders. So these studies were supervised with Professor Toru Minamino and Masayoshi Suda. He did extensive studies of GP and AV research. Thank you very much for your kind attention, and I'm happy to take questions.
Okay, thank you very much for your wonderful and so fruitful lecture, Dr. Shimizu. So we are now in the Q&A session. So yeah, we have a question here for the Miss Vika. So maybe we can, Miss Vika can answer this question first, and then we can continue to the next question. Please, Mrs. Vika.
Okay, thank you, Dr. Jeffy. Can I answer in bahasa?
Yeah,
or?
of course. Yeah.
Okay. Baik, terima kasih. First of all, I'm sorry, Dr. Sensei, if I answer in bahasa, because the question is in bahasa. Ibu Amana, terima kasih atas pertanyaannya. Mungkin untuk memperjelas dulu ya Bu, sebenarnya untuk epitope itu bukan untuk mengevaluasi immunogenicity, tapi kita bisa memprediksi immunogenicity dari epitope. Jadi mungkin agak kebalik gitu ya. Saya mungkin apakah seperti itu maksudnya ya Bu ya? Jadi kita punya epitope, apakah kita bisa prediksi imunogenisitasnya? Begitu bukan ya, Bu Amana? Ya, oke. Jadi untuk epitope sendiri kita bisa prediksi untuk imunogenisitasnya. Jadi bisa ke IEDB. Saya coba share screen-nya. Jadi ini adalah halaman IEDB yang tadi sempat saya tampilkan di PPT saya. Jadi untuk immunogenicity kita bisa prediksi di sini Bu. Jadi di IEDB dia punya resource, jadi kita bisa buka di sini MH. Tapi hanya untuk saat ini baru untuk yang kelas 1 aja. Jadi yang hanya ke HLA1. Nah, di sini kita bisa masukkan beberapa kandidat epitopnya, kita masukkan di sini. Kalau misalnya ada misalnya 10 ya, kita bisa masukkan 10. Ini sebenarnya bisa masukin example. Jadi kita bisa copy paste saja si peptide pendeknya ke sini, kemudian kita klik submit. Nah, nanti dia akan define angka untuk prediksi imunogenisitasnya gitu. Ini bisa di sini, kemudian kita submit. Nah, untuk skornya makin positif bikin imunogenik gitu Bu. Dan ini bisa ya, bisa diprediksi, tapi saat ini memang masih terbatas hanya pada MHC1 saja untuk resultnya gitu. Semoga terjawab ya Ibu Amanah. Sudah terjawab ya? Baik, terima kasih. Saya kembalikan ke Dr. Jeffy.
Okay, thank you very much. So, is there any question, maybe particularly for the Dr. Shimizu? Okay, actually, I have a question for the Dr. Shimizu. Maybe this is the important one. So, yeah, as I mentioned before, that several researchers would like to find the senolytic agent, like in the case of the drugs, yeah. But you, I think the idea was brilliant that you made the senolytic intervention as the vaccines. So yeah, I was wondering what did your purpose to perform in the vaccines format instead of the drug formats. Yeah.
Yeah. So at this moment, I think that there is no established method to deplete senescent cells. So one approach would be the exploration of the finding specific cell surface marker for senescent cells, but many other research groups are trying to do that, also testing like single cell analysis, but some of them mention that it's almost like impossible to find the cell surface marker specifically expressed in the senescent cells. But it's like a GPNB is a kind of one of the approach. So it's expressed in senescent cells, but not just in the cell surface, but also they have a endogenous expression is important for the homeostasis of the cells. So that will be one approach. So it continues important to find the cell surface marker expressed only in senescent cells. It may be different, even in the end cells, it may be different between the organs. For example, endothelial cells in the lung maybe have a different profile for endothelial cells in kidney or heart or whatever. So one approach will be if you can find, even among in the endothelial cells, if you can find a specific marker, we can, for example, specifically renew endothelial cells in the skeletal muscle, for example. So it's kind of an interesting research approach. But at the meantime, you're also interested in finding other chemical compounds or food materials that mediates a senolytic effect. And recently we identified, I don't know, it's a kind of hypothesis, but our study indicates that they utilize some of the already published senolytics, also our herb senolytics, specifically depleting senescent cells in a kind of cool way. So, so I hope our hypothesis, we can prove our hypothesis, but it seems that there is a kind of evolutionary, I'm not sure why it's evolution preserved, but we can activate the cell death signal only in senescent cells. So we're still extensively working on it. It's just started. So two approach is very important. Another approach will be not let the cells become aged. Let's keep them young, for example. So two approaches, and also two approach for senolysis.
Interesting. So yeah, is there any other question? So please don't hesitate to ask Dr. Shimizu and also Ms. Vika here. So while waiting the other question, actually I would like to adding my question.
I have a question for Jeffy.
Okay.
So is there any herb or whatever in Indonesia that use like a traditional medicine? Because for example, like China, they have many herb medicine, right? Many of them in the modern science cannot prove or even not tested to prove its efficacy. But some of them may be mediated through senolytic effect. So I don't know if you have, or if Indonesia have a kind of like a traditional medicine. Some of them may have a kind of a candidate to become to mediate senolytic effect. Just let the cubic cells senescent. You can radiate or put some doxycycline, whatever. Let them get aged. Just administer with several concentration. See some candidates may kill the aged cubic, for example. So in that case, you can say it's mediated senolytic effect. So is there any kind of, I don't know, traditional medicine in Indonesia or any herb or whatever? I don't know, it's just a comment. If you can find it, just do it, I guess.
Of course. Yeah.
We bring you some interesting results, I guess. I don't know.
Yeah, of course we have several herbs that some researchers also already analyze that these herbs have an antioxidant and the inflammation activity, but of course we should give further analyze whether these herbs also have some senolytic activity. Maybe like I mentioned, Dr. Wawemuli, as we discussed before that this drug, the Centella, so probably have the senolytic activity. Or maybe Dr. Wawemuli, do you have some comments?
Yeah, Mori Sensei, hi.
Okay, how are you, Sensei?
Very good.
Okay, nice to meet you. Yeah. Okay, so I just want to share information related to Centella asiatica. So in Indonesian terminology, we call it pegagan. So it is very common in tropical country like Indonesia. So easy to grown up. So actually in clinical setting, the Centella asiatica already make a formulation for topical preparation. So it will reduce the wrinkle. So in topical application, I don't know if we make an like drug to oral administration, whether it will be reduce the aging process. I'm not sure, but as a anti-wrinkle agent, so it is already proof in clinical trial.
I see.
Yeah, that's all.
So how many substrates are there altogether? For example, in our case, we are a little bit struggling to find whether there will be a single bullet or kind of combination of single bullets in the case of our herb, because there are nearly like 200 materials altogether making a herb. So we do not know which one, like a Yamanaka factor, whether there will be 4 or like combination of 100. We are still a little bit struggling to find a single bullet for that. So how about that one? Antarctica or something. Sorry, it's a little bit difficult for me to pronounce. Is it kind of a mixture of several?
So we can reduce the compound to less than 50. So
Really?
with a specific extraction process, so we just can extract only less than 50 compound that have benefit effect. Yeah, so we can exclude the harmful effect. So
I see.
I mean, Dr. Rafika maybe know how to extract Centella asiatica, so we can get the high content of beneficial compound. Yeah. So, but for topical, it is already proof. I'm not sure if it is by oral. Yeah. So we have to prove about the toxicity, something like that. Yeah.
Has anybody tested the senolytic aspects of the material? Is it senolytic or maybe no, I guess?
Oh, okay.
Yeah, we will discuss it later with you together with Dr. Wemuli, of course. So this herbs is one of our research project actually. So we will use these herbs, so we can use in the another purpose like the senolytic also. Yeah, and it will be interesting, Dr. Shimizu.
Yeah, but we have also to check the bioavailability because of the the flavonoid, something like that. The bioavailability is very low, so we have to increase the bioavailability by the extraction process to make it more lipid soluble, something like that.
I see. Sounds interesting. Yeah, I think there are several other candidates.
Yeah, actually, based on your herbs candidate, the Seno-H, so we know that maybe there are some myths that the herbs should contain the high flavonoid activity for giving the antioxidant properties. So is that the same condition in your herbs, that your herbs contains the high flavonoid, or maybe we don't need the flavonoid for giving the senolytic treatment? Yeah, what do you think about your Seno-H herbs?
Yeah, so we're still working on it. We don't have any knowledge about that. because there are 200 of them. So some of them are commercially available. So maybe we already tested like 10 or 20, but it's a little bit struggling to see conclude that one single food materials mediates senolytic effect. So it's kind of a combination, but it's kind of a really headache. So another point would be that suppose this herb includes like 200 compounds. All of them are not commercially available. So in that case, we need to collaborate with other research group to make produce the material itself. So it's a little bit a long road for us. So yeah, a little bit we're struggling to find, but the output seems to be robust. So the point is that suppose I'm a little bit speculative about the senolytic research, because at this moment, most of the papers say just show the reduction in beta-gal, show the reduction in senescent markers, like P53, P21, reduce, ROS, reduce. But still, I'm thinking, I'm a little bit about speculating. Maybe just it's simply because the ROS is reduced from the beginning, or it may have some role for suppression of cell senescence, showing the phenotype. So what I'm asking the lab members is continuously for like more than several years, asking them to do the xenofax. For example, as I told you, the SA beta-gal, a spider beta-gal, is a really easy reagent to detect the senescent cells. So okay, so if suppose we are interested in some drugs or reagents, whatever, senolytics, if it is a really senolytics, the senescent cells should show a kind of apoptotic markers, for example. So to clearly show the SA beta-gal cells, spider beta-gal cells should become positive for PI or annexing. So there should be like 3 combinations. So I'm asking. So it's a little bit easier to do that in the in vitro setting, but it's a little bit challenging to do in the vivo setting.
Yes.
We need to be a little bit careful when we say it's senolysis or suppression of senescence.
Yeah, of course. Yeah, thank you very much. So any other question? Maybe we have Dr. Imelda here from Physiology Department. So Dr. Imelda also performed several herbs, and yeah, maybe other participant for the other institution. So if you have some question for the vaccine, the herbs, for the anti-aging. So yeah, we got the, yeah. Do you want to give some comments directly, maybe Dr. Imelda, to the Dr. Shimizu?
Thank you for the opportunity, Dr. Jeffy. I'm really sorry I cannot open my camera. Thank you, Shimizu Sensei. It's really enriched my knowledge since I've graduated 5 years ago from Kobe University. So,
really? Okay.
Yeah.
Thank you,
yeah, he's my senior.
All right. Nice to see you.
Right now, we are trying to explore using several herbs as an anti-aging agent, and we try to establish an in vitro and also in vivo using rats and mice. So yeah, probably in the future, I will need your opinion or suggestion related
please
with.
Feel free to contact me.
Yes. So we will. I already prepare several grant proposals to the government. So hopefully, in the near future, I can get the grant, and then I can ask for collaboration with you. Thank you, Sensei.
Good luck for that. Yeah, we need a luck. Me too.
Okay, thank you very much, Dr. Imelda. So maybe any other suggestion?
Yes, thank you.
Okay.
Thank you.
So actually, I have another question, if mine, about the P53. So I saw in the Dr. Vika's lecture that you made the vaccine for cancer that targeting the P53. But as you know, also Dr. Shimizu, that the P53 can induce the senescent or the aging process. So it's kind of classic question actually. So every time I meet you, so everyone will ask, what if we suppress or we induce the P53, maybe in one side can inhibit the cancer, but one side can induce the aging. So maybe we can start from the Dr. Rafika first. So is there any future project, or maybe in the human, or maybe, yeah, what do you think that if we can apply the P53 vaccine in the human?
Yeah, thank you, Dr. Jeffy. Yeah, actually the idea of P53, because in cancers it's like neoantigens, neoepitopes that I mentioned before. For the maybe for the vaccine design, I'm not really sure about the how to implement the result for the design to in vivo or in vitro or in vivo, because from the paper that I read, need a lot of what's that? We need to make like models on the rats that I'm not really sure, because it's not my expertise. Yeah, maybe the difficulties to bring the design that we in bioinformatics usually use is to proceed further to experimental wet lab. So maybe with this discussion, if we have like a project further, we can collaborate. So bioinformatics can provide the design. We can give like a design to for peptide base to any target that we want to make. And for the next, maybe Dr. Jeffy and the teams can make the maybe the rat models to for the in vivo process. And for now, actually in bioinformatics, it's still difficult to to make it real. So we just designing and stop because we don't have any expertise for that. Yeah, maybe that's
Thank you.
Thank you.
Ms. Vika, it seems that Dr. Shimizu already answered the Dr. Imelda's question. So what do you think, Dr. Shimizu, about the role of P53 vaccine? What if we applied the P53 vaccine in the in vivo or in the human?
P53, mostly expressed in the nucleus, and also some of them are expressed in the cytosolic fraction. But I think it should be expressed in the cell surface. So suppose P53 extensively, mostly expressed inside the cell. I think it's a little bit challenging to target the depletion of P53. Yeah, so I don't know. It may not work as efficient compared with, for example, GPND or other vaccine targeting the cell surface markers for senescent cells.
So how about the P53 knockout mice in your previous research? Did that mice induce the cancer progression?
Yeah, that's right. So, for example, like a less than 6 months observation period, the mice have a kind of an anti-obese phenotype. But after 6 months, they start to die because of the cancer. Maybe they have kind of a lymphoma. So, for example, we tested systemic P53 depletion model and also adipose tissue specific P53 depletion model. They start to die after 6 months of the tumorigenesis. So not together with the other publication. So we also interested in not in the suppression of cell senescence, but the depletion of cell senescence. So so I received a comment from Dr. Imelda about the autophagy. So we are extensively studying the role of autophagy or organella together with xenometabolite we recently identified in humans. So we are collaborating with the biobank in Tohoku University. So we can analyze with a supercomputer for test the level of the metabolites in humans. So one of them seems to suppress autophagy, that in circulation in humans have a detrimental effect for the failing heart and also sarcopenia. So we are trying to establish the concept like a xenometabolite related disease that engulfing like Parkinson's disease, Alzheimer's disease, heart failure, sarcopenia. So I think it's recognized as a kind of, it may be recognized as one syndrome in the near future. So that's another research topic. I didn't share any data today, but maybe presented in the previous IMERI scientific meeting, I guess. The autophagy, as you know, it's a critical player for the regulation of aging and the aging phenotype.
Okay, thank you very much. Yeah, it's an interesting topic actually, targeting the senolytic by herbs, by vaccine, also the conventional therapy. Yeah, but unfortunately the time is limited, so the time is already running out. So maybe we can finish this discussion. Thank you very much for Mrs. Rafika and also Dr. Shimizu. Yeah, surely I will contact you soon to further discuss the herbs. Yeah, thank you very much.
Thank you very much.
Yeah, we will have another webinar series in the next April for the anti-aging, the diet intervention. So yeah, this we can attend also in the next session. Anyway, thank you very much, Dr. Shimizu, and also Ms. Vika, and also other participants for your fruitful discussion. Thank you, and see you in the next session and the meeting. See you.
Okay.
Bye-bye.
Thanks, JP.
Thank you very much. Yeah.
Thank you. Bye-bye.
Thank you very much.
Okay.
Okay. Thank you.